Pleasure to have Professor Meir with us today. Did his PhD in the Weizmann Institute, and then he did a postdoc for 2 years in Caltech. He works extensively in the field of machine learning and shifted his interest in recent years to theoretical neuroscience. And he's currently a faculty in the Department of Electrical Engineering in Technion. And today we'll hear his talk on the neurons' response at extended timescales, memory, noise, and stability. Thank you.
Okay. Okay, thanks. So this talk is essentially going to be based on the PhD work of Daniel Sudri, who completed his PhD recently and actually left 3 days ago for a postdoc at Columbia University. So the title, I've shortened it a little bit because I don't think I'll be reaching the topic of stability here today, but I will discuss extended timescales and the issue of memory in neurons. So let me start with some motivation for looking at extended timescales in neuroscience in general. People often refer to long timescales, and they usually attribute it to higher levels of functioning, such as behavior, maybe systems level, network levels, and one usually associates some kind of emergence phenomena associated with having started with short timescales and having interactions, complex interactions, leading to very long timescales. However, long timescales often exist even at very low levels, be it the level of ion channels, where timescales have been observed to take place on scales of minutes and more, and neurons. Recent experiments from Shimon Marom's lab, which I will refer to when you heard the talk by Shimon a few weeks ago, are going up to scales of days. So the basic question we are asking in this research is whether generic biophysical models can actually explain these long timescales in any fundamental way. So the basic question one can ask is essentially, what does a cortical neuron do? Now, of course, that is not a well-defined question, so let's try to unpack it a little. If we consider a cortical neuron, or any neuron for that purpose, if we're looking at a pyramidal neuron, we're talking about a neuron which is receiving maybe a couple of thousands of inputs and outputting some kind of spike train. And specifically, in engineering, one usually asks, what is the input-output relation of this system? And this is a system, so one can try to ask, what is the input-output of this system? The question is, what is the input to the system? It's not exactly clear what we mean by the input to the system, and we'll try to explain what I mean later. Now, one difficulty is that it's really impossible to simultaneously measure. No one is able to measure all the inputs into a neuron and the outputs and do that simultaneously. And the question we're asking here is, how far can biophysical constraints take us in trying to explain this input-output relation of a neuron? Now, although I've depicted here a neuron with its dendrites, etc., I'm going to focus on a point neuron here. Even that is probably too complex for full analysis in mathematical terms. Now, the framework I'm going to work within here is that of conductance-based models, and this is a very well-known framework in neuroscience, originally attributed to or developed by Hodgkin and Huxley, who developed the basic formalism for this kind of models, and arguably it's probably the most successful mathematical model in neuroscience. Now, it's been very useful over the years for understanding intracellular dynamics. It's less popular when one goes up to the levels of understanding response to stimuli or to network models. The difficulties here are that it's a highly complex nonlinear model with many parameters, only few of which are in general identifiable. So I've depicted here one type of ion channel, calcium, a model, a Markov model for an ion channel, and even this is highly complex. Adding onto that the nonlinear dynamics of the voltage, you very quickly run into extremely difficult problems which severely restrict the type of analysis or mathematical analysis one can form on these systems. And again, this issue is a difficult one because many of the parameters of these models are not really identifiable. So, to make things worse, even if you look at the most detailed conductance-based models, which are described by maybe 30, 40 nonlinear differential equations with various types of ion channels, even these are highly simplified models of reality. And moreover, most of them assume, or many of them assume deterministic. The Hodgkin-Huxley equation is a deterministic equation which basically assumes that the number of ion channels is large enough that some kind of law of large numbers applies to theirs and averages out their stochasticity. One assumes homogeneous synapses, and usually these models are use only a very small number of kinetic processes, whereas we know that the new types of kinetic processes are being discovered daily. So these complications can be, we've known for a long time that ion channels, a single ion channel is a stochastic element, a stochastic system in terms of its current voltage relationship, and these are just the description of a current flowing through an ion channel, and it's highly stochastic. And moreover, work by several people has shown that the distribution of synaptic efficacy, synaptic strength, has this kind of long tail distribution, so that you have a few very strong synapses on top of a kind of sea of relatively weak synapses. Now, in this paper by Kagaya et al., they claimed, and I know this is a controversial issue, that a few presynaptic spikes can actually evoke a postsynaptic spike on top of a kind of a noisy background of the weak synapses. So the situation here is very different from the kind of situation one often assumes, where you have a sea of relatively homogeneous inputs impinging and the some kind of law of large number effect of all these input currents affecting the neuron in some kind of mean fieldish kind of way. And the situation here seems to indicate that the nature of the inputs is not what we assumed it to be. Now, as I said, all these models have always had some kind of, although they are very complex, new processes are constantly being discovered at various new types of channels, new types of interactions, new types of effects of interactions, especially at long time scales. So for instance, ion channel regulation, modification of the density, for instance, of ion channels, the changes in the concentration of ions, and actually even changes in the spike initiation zone when you look at neurons on scales of many hours. So at these very long time scales, a lot is going on. And the question is, what can one do in trying to understand neurons at extended time scales if one takes into account these, endless number of complexities. Moreover, as Steriade in 2003, he has a paper about the flexibility of cortical neurons, and he says the same neuron exhibits several discharge patterns that are otherwise ascribed to different neuronal types. So you can have a single neuron, and at different times in its history, in its lifetime, it has different types of behaviors. So if you think of it as an input-output system, I mean, if I look at a transistor in the computer, its input-output relation will always be the same. However, if I look at a neuron at different times in its history, depending on its history and on the current environment, it may behave very, very differently. And even at the level of ion channels, although most models of ion channels are non-Markovian, essentially recent models of ion channels describe them in non-Markovian senses as channels that have a very long history, because we know from work of Shimon Marom and other people who have looked at ion channels that these channels are not best described as Markovian systems. So the usual alternatives to going into all this kind of endless complexity is to use kind of statistical models like generalized linear models, which have the advantage of being very, very easy to fit, and the disadvantage is that they're really completely unconstrained biophysically, and they are usually applied for relatively short, or applicable for relatively short times. Other kinds of models are simplified conductance-based models, which are like integrate and fire models or quadratic integrate and fire models, various models of that nature, which again are relatively easy to fit and analyze, although even they are not always trivial to analyze. Again, they're not really biophysically constrained, and it's not always clear when they are oversimplified and what kinds of phenomena they can explain. So one thing I'd like to point out, and I'm sure everyone here, including the young students, has seen this figure from the work of Senofsky and from the review of Senofsky and Churchland, which, and the reason I'm depicting this is that a neuron, essentially, if you're looking at a neuron, it works within a context of a whole system starting from molecules, synapses, and all the way up to the CNS, and actually up to higher levels of the body and behavior as well. Now, when one usually looks at this kind of picture, one has in mind a kind of feed-forward mapping from molecules. This is a very physics-like picture where you have emergent phenomena taking place at higher levels, essentially due to bottom-up kind of interactions, going from molecules to synapses to neurons, etc. However, in reality, there's a lot of feedback between all these levels. First of all, there is the level of behavior, which we should put up here. And then there are interactions all the way down. For instance, a very well-known interaction at the level we will talk about is between neurons and molecules. For instance, the ion channels and the voltage, the voltage of the neuron, which is essentially a collective effect of many ion channels, feeds back obviously onto the channel level. You have feedback from the network to the neurons. You have feedback from behavior through learning on synapses. So really, when you're speaking about a neuron, you have to look at a neuron in the context of its overall operation. Now, neurons in general are able to operate for extended periods of time, for months or years. So the question is, what happens to these neurons over this time, and how does their behavior change across these multiple time scales? So to summarize this maybe too lengthy motivation, one problem is that these conductance-based models are too complex to use. There are many parameters, essentially mostly unknown. They're highly nonlinear and difficult to analyze mathematically. On the other hand, they're obviously too simplified to be realistic. At least some reasons for that is the issue of stochasticity, which has often been neglected. There is this issue of temporally sparse inputs, that actually a small number of inputs may actually affect the neuron at relatively sparse temporal points. And all these slow kinetic effects, which are being discovered on a daily basis, really complete add-on complexities to the system and really prohibit any kind of general understanding. So the point that I will try to make here in this work is essentially that to a large degree, problem 2 essentially solves problem 1. And I would say that the correct way as far as I see it, to understand a neuron now is essentially as a network, as a population. A neuron, from my perspective right now, is a population of ion channels. And actually trying to understand the properties of a neuron are, in a sense, an emergent property of these ion channels. Exactly as a network, the properties of the network are an emergent function of the neurons in this network. So let me start with some brief experimental motivation. And again, Shimon Marom talked here a few weeks ago, so I'm sure he discussed this much more extensively. But most of the motivation for this modeling work I'm discussing here is coming from the work they did at their lab, which is based on a culture of rat cortical neurons in a multi-electrode array. And what they were able to do is to isolate a single neuron from all synaptic inputs. So they really are able to study a single neuron across extensive timescales up to 50, 60 hours. They use a fixed amplitude extracellular current pulse stimulation. And initially, in the initial work they did, and they later extended it, the stimulus was periodic. So the situation they have in this experiment is that the neuron is stimulated periodically, and one looks at the response, at the action potentials produced by the neuron. So there is usually a slight latency between the stimulation and the response. But what is interesting here is that the neuron, in certain regimes, and I will discuss this in a moment, sometimes it responds to the stimulus and sometimes it does not respond. So there is some kind of intermittent effect where the neuron is not responding on a constant basis, but sometimes decides not to respond. So what they did here is in this experiment, they look at periodic stimulation. And if you start with a very low stimulation frequency, maybe 1 hertz, the neuron responds in a stable one-to-one response pattern. So for every stimulus, it responds. Then at a fixed latency. Then as the frequency increases here up to 20 hertz, the neuron starts going through this transient phase where it still responds to every stimulus, but the latency of the response keeps increasing. At some point, the situation breaks down, and you have this so-called intermittent regime where you sometimes get a response, sometimes there is no response, and the latency actually fluctuates, the latency of the response. And then when you reduce the frequency of stimulation back down to 1 hertz, things go back to their initial phase. So in previous work, we actually used deterministic models to understand these regimes, the stable and the transient phase, but I'm going to focus now on this intermittent phase and try to explain this issue of action potential failures and why, and what is the mechanism driving that. Okay. So just to describe that mathematically, if we're looking at a stimulus which is based on a periodic stimulus, so these red arrows represent the stimuli, and you get responses which I will denote by Ym. So let's say Ym is 1 if there is an action potential and 0 if there is no action potential. So essentially, you get the sequence of zeros and ones. You get some long binary string of zeros and ones. Responded, did not respond, responded, did not respond. And you could go on and you can fold them on. And what Asaf Gal did in Shimon's lab, you actually, across 55 hours, you can just plot the black and white dots depending on whether the neuron responded or did not respond. So what you see when you analyze, or even if you visually inspect this figure, you see a high amount of variability, irregularity, and actually, if you quantify the effects that are taking place here, you see multiple timescales in the responses, and interestingly, you see this 1/f type of spectrum. Now, I remind you, the spectrum essentially is just the Fourier transform of the correlation function. So what you see that the spectrum at low frequencies has this kind of 1/f. Essentially, it's a 1/f to the alpha type of behavior, where in their experiments, alpha was around 1.5 before, and you see it is quite variable. So there are obviously different experiments, you get different numbers, but this is the kind of typical value. So, as I said, what I will try to do is try to explain these phenomena in the context of conductance-based models. Now, let me just go through a brief reminder of the Hodgkin-Huxley model, and I'm sure most of you have seen this model. So the Hodgkin-Huxley model is essentially a model of which describes an electric circuit with resistances and capacitance, where the resistances or the conductances are nonlinear functions of the voltage. So if you look at the sodium current or the potassium current, they are given by these functions here, where each one of these variables, m, n, and h, which are the gauging variables of the different ion channels, obey this kind of differential equation, which is essentially a kinetic equation, where the alpha and beta are the rates for these kinetic processes. And again, the Hodgkin-Huxley is, of course, as it stands, a deterministic set of equations. The original model had 4 equations. These are highly nonlinear equations, which are even for this very simple original 4-dimensional Hodgkin-Huxley, there is no analytic solution to that equation. Only under various reductions can you solve it exactly or even characterize the solution. So, however, we know that these kinds of, this model and its extensions, they have this kind of excitability property. So what do I mean by that? If you're looking at the various parameters characterizing the situation, maybe the stimulation amplitude for a fixed periodic stimulus, and maybe some variable related to the initial conditions. So there is some kind of physiological range within this neuron, within which the neuron operates. Now, there is a region within this physiological region where you get action potentials, and there is a region where no action potentials are generated. So when you have no action potential, maybe you give some stimulus, which causes some subthreshold dynamics, but no action potential is generated. Whereas in other situations within this green region here, action potentials are generated, and this is, of course, something all of you know very well. Now, again, mostly when one looks at excitable systems, one usually assumes, and this is not strictly mathematically correct, but is correct to a very good approximation, that action potentials really their shape does not depend very much on the initial condition and the stimulation amplitude, that they have a rather stereotypical shape. And of course, this is not exactly true, even in the Hodgkin-Huxley model in certain situations, but this is the standard kind of assumption. So over the years, people have realized that the Hodgkin-Huxley model, which was originally constructed to explain the behavior of the squid neuron axon, only is restricted in many ways, and over the years people have gone on, added various types of kinetic processes like sodium slow inactivation, potassium slow inactivation, and you can go on adding more and more, and over the years these models have become increasingly complex, and people have developed more and more elaborate models, again going up to dozens of types of kinetic processes. And what one usually does with that is one puts these equations into a computer and just solves them numerically. Now, the assumption that will be underlying this work essentially is that there may be multiple types of kinetics taking place here. However, there are basically 2 types of kinetic processes which we can deal with, or 2 types of variables. We have the rapid variables like m, n, and h of the Hodgkin-Huxley model, which are essentially responsible for the actual relatively fast dynamics of the generation of the action potential. And these usually have timescales on the order of milliseconds, maybe a little more. However, on top of that, there are slow variables which are related to the action potential modulation, to the excitability, which are actually, and they affect, they may affect the action potential shape and rest voltage, but on a much slower scale. And we will refer to these as R for rapid variables and these as S for slow variables. Okay, so in general, as I said, the Hodgkin-Huxley model is a deterministic model, and the question is, what do we do when we want to take into account the fact that ions are stochastic elements, ion channels, sorry. Now, usually what one does is one models these ion channels as, for instance, this sodium ion channel in terms of these Markovian state models. Of course, I should add that there are other non-Markovian channel models. But the question is, how do you incorporate these kinds of models into a Hodgkin-Huxley formalism? So obviously you can do that. You can just run the system and you can run a finite number of ion channels, and each ion channel you can simulate the Markov chain. This Markov chain may be using Monte Carlo methods, and obviously this is, first of all, from a mathematical point of view, it's not very easy, to say the least, to analyze that, and actually it's also very time-consuming to simulate them. So over the years, since the original work on Fox and Liu, various people have tried to translate these kinds of Markov chain models into a system, into a differential equation systems. They're called Langevin equations. That's not very important, which are rather generic descriptions of stochastic conductance-based models, where you have the equation for the voltage, which may be similar to the equation in the Hodgkin-Huxley model. And then you have stochastic differential equations for the variables R and S. Now, in this case, you can interpret R and S essentially as the number of open channels of a certain type. So since they're the number of open channels, they are random variables because we know that the behavior of the ion channel is stochastic, and whether it's open or closed is some kind of a random variable. It's the probability, and which has some kind of probability associated with it. So I'm not going to go into these equations. These are going to be the starting point. These are work by other people. Essentially, what you have here is a deterministic component of these equations, which is just a linear kind of equation, plus some random stochastic noise process where the matrices BR and BS are related to the number, the order of magnitude of these variables is related to 1 over the square root of the number of ion channels. So the more ion channels you have, if the number of ion channels of a certain type goes to infinity, these terms are absent and you have a deterministic system. So if you have a finite number of ion channels, you have to account for the stochasticity of the ion channels. And psi are just random noise, white noise. Okay, so this is the equation again. Now, the usual, when one looks at models for neurons, one often tries to understand the relationship between the input current and the voltage. Now, this is usually a very hard problem because these systems are highly nonlinear, and if they're stochastic, they add onto it the difficulty of dealing with the stochasticity. And these are, in general, trying to understand the input-output relation for a general conductance-based model is in general quite intractable. So the kind of approach we're trying to present here is essentially to use the sparsity of we're going to make an assumption that the input is temporally sparse. We're going to use the kind of excitability properties of the neuron and try to be able to say, to provide some rather general results for a broad class of conductance-based models. So there are many types of models which people have used over the years, and this is just a small sample. HHS is just taking the original HH model, Hodgkin-Huxley, and adding slow sodium inactivation. There are models which add multiple sodium currents, or this model which essentially is the original model plus inactivating potassium currents, and you can go on and you can look at an endless number of types of models, and people have actually done that over the years for describing this voltage dynamics. So what we're going to try to do is present some kind of unified analytic formalism which is applicable to all these kinds of models. Of course, as always, when we make such claims, we're going to have to make some assumptions, and some of these assumptions may sound rather strict, but I will try to show that they are not as strict as they seem. So, okay, so the setup we're considering here is you have a set of input stimuli which are essentially very short pulse stimulations, and we're looking at the response. Now, you see the input does not have to be periodic in this formalism. So we have these stimulation points. The T1, T2, etc. Define the intervals between the length of the intervals between the stimuli. And again, Y1, Y2, etc. Describe the yes-no existence of an action potential. So the assumptions we're going to make here is that the inputs are temporally sparse and that no spontaneous action potentials are generated. And these are two essential assumptions for this formalism. The first, what do I mean by temporally sparse? I mean that the time scale of the action potential of the Hodgkin-Huxley model of the generation of the action potential is much smaller than the time scale between the stimuli. So this is one assumption. And there will be a second assumption which I will state shortly. Now, the objective would be somehow, based on these kinds of assumptions, to be able to come up with a general relation, the general mapping that maps the input. So I view the intervals. You could view the actual times of the input spikes as the input, but it's more convenient for me to define the intervals between the input stimulation, between the input stimuli as the input variables, so T1 to Tm. And I'm trying to characterize this relation between the input and the output of this neuron. So in general, because of the temporally sparse nature of this stimulation, one can actually easily convince oneself that you can describe this kind of dynamics in a Markovian kind of setting where the xi's represent the state of the neuron, that is the state of all the ion channels, all the terms which determine the action potential. And then the response is just this binary variable. So you have this kind of, the internal dynamics of the neuron is taking place on this level above here. And at each time point, when you have a stimulation, at these intervals, you either get or do not get an action potential. And whether or not you obtain an action potential, this influences the internal state, because we know that the state of the ion channels is voltage dependent. So whether or not you have an action potential, of course, it affects the internal state. Now, one could actually say, well, okay, all I need to do is I have to characterize the dynamics of these internal variables between these mapping points and the dynamics of the output variable, the probability of spiking, and then I can actually come up with the most general description where, given the inputs, I can compute the distribution of output spikes.
You have such distribution for every stimulus strength? Because here is only
yeah, right now I'm assuming here that we have a fixed stimulus, but obviously you will have this, it depends on the stimulus strength, sure. So computing this in general is intractable, and this is where we will have to make a certain assumption which will actually allow us to simplify the analysis. So these are the equations again. So as we previously had this assumption stating that the time scale of the action potential was much smaller than the intervals between the stimuli. Now I'm going to make a further assumption, which is many of you are not going to like, and this is that the internal time scales of the slow variables are much larger than the intervals between the stimuli. Now, As I will show later, this assumption, I have to make it for mathematical reasons, but it turns out that the results, and I will show that, actually are applicable even in cases where this assumption is not strictly true, although we cannot mathematically show that it holds in that case. So in this case, essentially what you have is you can replace the previous diagram with a similarly looking diagram where what appears here, instead of the full set of internal variables, just the slow variables. So you have these slow kinetic variables which are evolving on a relatively slow time scale. And they are affecting, and the fast variables are essentially what determine the appearance or lack of appearance of an action potential in this. So you have a slow time scale up here and a faster time scale up here. And we make this assumption of this separation of time scales. Now all the arrows here, of course, describe stochastic transitions. So without going, and I'm skipping all the mathematical details here, what one is able to do under these assumptions is to show that for and I apologize, there will be a few hopefully not too lengthy equations, but you don't have to, I'll try to explain what they mean. You can actually replace these stochastic differential equations which describe the dynamics of these neuron models by a discrete time mapping with an index m, which describes the state of all the slow variables. Again, the slow variables are all the slow kinetic processes. Ym is this term which tells you whether or not you obtain an action potential. And what you see here is that Ym appears here and 1 minus Ym appears here. So what this means is that the equation is influenced by, if Ym is 1, that is, an action potential occurs, you update the slow variable in this form, and when an action potential did not occur, you update the equation based on this term. Now, these terms Nm and Em are noise variables, are white noise processes, which are related to the stochasticity in both the generation of the action potential and in the dynamics of the slow variables.
Why does time have to be discrete? Why can't it be continuous?
Well, because within this, time is continuous, but within this formalism, I'm talking about a situation where you stimulate the neuron in discrete time pulses. So what one can do in this case is one actually can translate the continuous time description into a mapping which just maps every time you have a stimulus, you update your parameters. So you don't, these equations, these H pluses and H minuses, essentially sum up all that is taking place between the stimuli. Okay?
So M is not time, it's the stimulus.
Sorry?
M is not real time, it's the stimulus.
Right. So yeah. So M is the time of the stimulation. Yeah. So this is a kind of a formal mathematical relationship. However, each of the variables here, H plus, essentially the H variables and the N and the E are biophysically interpretable. So given your specific favorite conductance-based model, we can actually compute these functions for that specific model and tell you what they are. Of course, this model has far fewer parameters than the original conductance-based model, and in terms of simulations, it's much faster than actually simulating the original model. Now, one further point which one can notice in order to make the system, this is a nonlinear update equation. However, in situations where where the input is a random process, essentially if you look at the PAP of Sm describes the probability of generation of an action potential given the state of all these slow variables. Now, if the dynamics is stochastic, you can actually assume that the Sm reach some kind of asymptotic fixed point, and you can expand around this fixed point, and you can actually, this is usually a sigmoidal kind of function, and you can actually linearize it. And so this is an approximation we're going to make because it simplifies the analysis considerably. And as you can see, now this is only true for, this is in a sense, we can only do this linearization because the system is stochastic. If the action potential generation were deterministic, in previous work we proceeded along a different route. But in this case, where you have stochasticity here, you can actually linearize this relationship without losing too much accuracy. So let me describe the main kind of mathematical results. So again, this is the setup here, and I'm going to denote Tm, again, with the intervals of stimuli. So I'm going to assume they're given by some average term plus the deviation from the average, and the same for Ym. Okay, so the average value plus some deviation from that. So what we can show essentially is that this kind of, this system, we can actually fully describe it in terms of two mappings. The first is a nonlinear mapping of the mean value. So there is some nonlinear mapping between the mean value of the input intervals and the output variable y, and this is some function which is related to the probability of generation of the action potential. And these deviations, these stochastic variables, t hat and y hat, are given by some linear mapping of this form, where here you see, now I apologize to all of you who have not seen engineering type block diagrams before, but what you have here is a linear system where you have the input coming in, going through some kind of what is called transfer function and generating the output here. And this upper plot here describes some random independent white noise process going through another filter. So essentially we can fully describe the dynamics of these hatted variables in terms of a linear system, linear stochastic system here. So,
sorry, can you just go back to the justification of linearizing?
Yeah, so in principle, this is a nonlinear function. Now, this nonlinear function is usually some kind of sigmoidal function. So now, if the system were fully deterministic, this would be some kind of threshold function, and obviously you couldn't linearize it. Now, what I'm saying is that assuming that this slope is not too steep, we can linearize it around some fixed point S star and replace this function by some kind of linear function. There is no deep mathematical justification for it. The justification I will provide is it facilitates the analysis significantly, and as we'll see in the end, it provides very, very accurate results. So I cannot prove mathematically that it's justified. Okay, and what is interesting here is that this formalism allows us to separate the inputs. So the input-output mapping of the intervals t to the output variable y is fully specified by this function h signal, and all the noise processes resulting from the stochasticity of the ion channels, all that is pushed into this upper level here, and they are totally separate. And this is the main reason that we are able to do that is this linearization process. So essentially, and I apologize, the next few slides will have a few equations, but hopefully they will not be too painful. Again, we're trying to map the input intervals to the output variables, the spikes. So what we can essentially show that under the approximation that I've used, essentially you can write down a linear map for the slow internal variables f, and a linear map for the output variable y hat. Now you see these variables f, d, a, w. All these variables are completely derivable from the specific conductance-based model you're using. So you can actually compute them, and this is the most complex figure here, so I will essentially what I'm saying is the following. You're trying to map the input variable t, the intervals between the stimulate, to the output variable. So there is a mapping between an input and an output, and this mapping is taking place through the slow internal processes of the neuron, which essentially represent all the slow kinetic processes. Now, the slow kinetic process affects the generation of an action potential here through this variable w, and the action potential itself affects the slow variables through this term here. So you have the slow dynamics, you have the relatively fast dynamics, and then you have these noise variables n and m, which are affecting the system. So this is a fully linear model which describes, again, the deviations, the hatted variables, or the deviations and the mapping of the input-output of this system. Now, again, I emphasize that all these variables in this model are completely derivable from the biophysics. So any conductance-based model you give me, I can compute these variables. Now, what is interesting, and this I think has an important philosophical message, is that there's a huge redundancy. And I think Yves Marder in recent years has stressed this redundancy. There is no single parameter describing a neuron. There can be many, many multiple parameters at the microscopic level, which will all give rise to the same input-output relation of the neuron. So as long as these D, W, F, and A variables are the same, many types of models can lead to the same kind of... so there is no correct model in
the case of these models, they have the same fractals. They don't have the same dynamics. They don't have the same transient dynamics.
In the case of a neuron or of a network?
Network.
Yeah, but I think she has also made such claim in the case of neurons, and I'm not sure. They may not have the same dynamics. I'm saying these are what I'm referring to essentially are the statistical properties. This is a stochastic mapping. So what is more relevant, and I will get to this, is actually the statistics of the mapping is the same. Like this, if the spectrum, for instance, of the output is the same. So I will get to that in a moment. So from this kind of analysis, one can derive the mean firing rate, the second order statistics. You can actually compute optimal linear filters, and you can actually, it's much easier within this kind of model to do system identification, because if you're looking at a Hodgkin-Huxley model or some variant of that, which has maybe 55 parameters, and you're trying to estimate these parameters from the data, that is usually quite impossible to do. So one quantity which Asaf Gal in Shimon Moron's lab has looked at in terms of his experiments is the spectrum of the output spikes, and that is something which behaves in this kind of 1/f nature. So from this formalism, we're able to come up with a closed-form analytic expression for the spectrum. Now, I apologize in advance for the next equation. I don't expect you to comprehend it. It's just there because I want to show you that we have a closed-form expression, and then I will show some figures describing what is going on. So, actually, this expression, which I already apologized for, gives the closed form spectrum of the spikes of the output depending on the statistics of the input. Now, again, all the parameters are determined from biophysics. Now, the question is whether, under what conditions, this kind of spectrum can display this 1/f to the alpha type of behavior that has been observed in Asaf's experiments.
This is the linear model spectrum.
Right. This is the spec.
What type of noise do they have?
This is for a general. Sorry. Yeah, for this expression. 1 second.
Yes, stochastic input.
Yeah, it's stochastic input. I don't recall exactly what the assumption was on the input for this. Yeah, but I mean this. No, as far as the statistical properties of the input are already embedded in that equation. The variables A star, etc., and D star already contain the statistical structure. Only second order moments, of course, of the input are in there already, because this is just the output spectrum. It depends on the input and the transfer function. Okay. So the question is, if you have what type of conductance-based models lead to this kind of to 1 over f type of behavior in the output, and then assume that you are able to generate this 1 over f type of behavior in the output spectrum. What is the nature of the input-output mapping? So okay. And then the question is whether we can make any kinds of predictions related to, suggesting some experimental question. So the procedure we take here is we choose the model parameters for this linearized, for this effective model, which leads to a spiking spectrum of 1 over f to the alpha, and I'll show you that this is possible in a moment. And then this leads to a certain specific scaling in the number of ion channels within our model. Then we look for some experiment, for instance, with some value of alpha equals 1 over 4, which was observed in their experiments. And then we will compute the other types of statistics for the model and try to compare with the experiment. So, following quite extensive analysis, one can come up with some rough criteria for generating 1 over f to the alpha types of behavior in these models. First of all is that this will only work if you have a finite number of ion channels. So at least within this formalism, noise seems to be essential to the generation of 1 over f. We need to have some slow processes, some slow kinetic processes, which have a time scale which covers the range over which 1 over f type of behavior is observed, and that slow processes are actually noisier. What do I mean by noisier? That slow processes there are fewer ion channels with very, very slow processes, meaning they are noisier. So we can look at a very simple model. This is the general kind of model. A very simple model is where you have uncoupled ion channels, where each ion channel has this kind of behavior. Now, assuming that the rates, the gamma and the delta here are the transition rates, that they have this kind of scaling behavior for some parameter epsilon. So you have exponentially scaled rates for different ion channels. And this is just one example. And in this case, we can predict that if the number of ion channels at a scale k behaves in this kind of fashion, where alpha is this parameter here, so we predict a specific that slow channels are effectively essentially noisier. We have fewer and fewer slow ion channels, and under these circumstances, 1 over f type of behavior will be obtained. And again, within this model, we can actually show that if there is no stochasticity in this model, deterministic model do not generate 1 over f behavior. Now, I'm not making this as a categorical statement, and actually, Shimon and Asaf have a model which generates 1 over f behavior. Right. So I'm saying within this model, I'm not saying it's categorically impossible, and I will get back to that at the end. Okay. Within. Yeah, but this is a very specific system, right? And
on the other hand, a linear system, any finite dimensional linear system, is only Gaussian noise. It will never generate.
Okay, so I mean, this is actually an issue I'll get to back to at the end, and this the reason I put this star here is that again, this is not. It may very well be due to the linearization I've made. Okay, so I'll get back to that. Now, just to show you that this analysis actually works, what we have here is, for one of these models, which is this HHS model, we plot here the spectrum of the output spikes. So the blue is the actual simulation of the full HHS model with the stochastic ion channels. The red is this discrete map I described, and the green is this analytic approximation which I showed you, this complicated function which I showed you. And you see that in both cases I can, and this is the spectrum of the internal variable S, and you see they are very, very well approximated within that equation. And we tried various other types of models, multiple models, even a model which does not exactly fit our assumptions, where the inputs are coming through some kind of adaptive, slow, adaptive synapses. And you see that in all these cases, this kind of exact closed form expression for the spectrum provides a very nice description of the structure of the spectrum. Now, actually, it's interesting to see what happens when the assumptions fail. So this is for one specific model. For instance, here we see a situation when the mean interval between the input stimuli is 0.5 tau s. Now, this contradicts our assumption that the interval is much larger. Sorry, that the tau s, the time scale of the s variables, is much larger than t. And you see that still, although our assumptions are not obeyed here, you still see a good agreement. And this is, again, a situation where the assumption of a fixed point fails. And again, we can pretty well describe these systems, even in cases where the assumptions fail. So, of course, I don't have an explanation for this, but it's encouraging to know that the assumptions are not that strict. I think for the lack of time, I will skip this figure, which essentially shows that all the measures that Asaf Gal used in his paper to demonstrate the scale-free behavior of the neuron they were looking at essentially are reproduced within within our model, and there are multiple types of variables like the Fano factor, the distribution of intervals between responses, etc. But I will skip this. It's a rather complex figure anyway. One issue to look at is actually to do system identification, because now we have a very simple linear kind of model, and we can use standard linear estimation techniques, maybe a Kalman filter, maybe ARMA models, what have you. And we can try to predict, given the inputs and the previous outputs, whether or not a spike will occur. And what we see here is that the x-axis is the number of ion channels going up from 10 to the 4 to 10 to the 12. And the blue plot is just some trivial prediction based on the mean. And the red and the green are essentially predictions based on our model or based on an oracle which knows the internal state of the neuron. So essentially what you can see here is that we are able, obviously you see that for relatively small number, even large number of ion channels, 10 to the 6, the neuron is very, very noisy in terms of it's very non-predictable. As you go to a larger and larger number of ion channels, the neuron becomes more and more predictable. In a similar way, we can try, based on the observation, to estimate the internal state. And again, what I show here is the actual internal state in blue, and the estimate based on a prediction of these internal variables in S. And you see again that they agree quite well, in spite of the various approximations we've made. So let me just try say something about the input-output behavior of the neuron. So I talked about the spectrum, and what is really interesting is trying to relate the input of the neuron, that is the temporal events, to the output, that is the spiking events. Now, within the model, within the framework we have been looking at, you can show that there is a simple linear relationship between the input and the output. This describes the direct dependence on the input, and this variable describes all the internal fluctuations, all the ion channel noise that exists in the system. And so if you're looking at this in terms of the Fourier space, you can actually separate the output into two components, one which results from the input and one which results from all the internal noise processes. And what is interesting here is that if you look at the time scales of the response, the filter which filters the internal noise, that is if you look at the correlations that result from just the internal noise, the ion channel noise, it has this kind of shape, and this holds over time scales of days. However, if you look at the time scale of the response to the inputs, has a time scale of maybe 100, 200 seconds, time scale of a few minutes, and it has this kind of low-pass filter shape. So what this essentially implies that within this framework, the the output of the neuron essentially has a memory which can remember its input on a time scale of minutes. Okay, a minute, 2 minutes. And interestingly, earlier this year there was this paper by Pozzaroni and Gerstner and other people which looked at simple kind of generalized integrate and fire models, tried to fit them to data, and came up with these kind of, again, input-output filters for the data they were looking at, which were fitted within these models, and discussed noted that the input-output memory is on a time scale of 20 seconds for these for the data they were looking at. So we're talking of the same order of magnitude, maybe not 20 seconds, and the shapes of the filters again are this kind of low-pass filter shape. So I'm running out of time, but I will just say, okay, 2 minutes, that should be okay. So the point is that we can actually try to probe directly. Remember, H external is essentially the mapping, describes the mapping between the input and the output. So if we're looking at the correlation between the input and the output and taking the Fourier transform of this correlation, which is just this function, we can actually estimate it from the experiments done by Asaf Gal, and they did experiments specifically for input statistics, which was a 1 over f to the beta process, and we can estimate the SYT from our model and compare it. And what we see, so this was the 1 over f kind of input statistics they used. And again, based on the models we fitted to the spectrum, we tried to directly infer the input-output mapping, this H external function. And again, comparing with the experiment, we see that we get very much the same kind of behavior in terms of the correlations between the input and the output. Okay, one last example, and I will just discuss this very briefly, is that because of the approximation we've made, we're looking at a linear system, and you know, hopefully, that for a linear system, if you take a sum of sinusoidal functions and you input them to a linear system, you get a sum of shifted and scaled, rescaled sinuses. So, what we essentially, so this is this kind of statement in equations where if we have a sum of sines at some frequencies FL, the output of the system is also going to be this kind of sum of sine functions. So what we can do is we essentially estimate this, this mapping, this input-output transfer function based on the peaks of the Fourier transform of this function, which will occur at the frequencies FL. So basically, you can use this, and we're actually suggesting a new experiment, which has not, there was some preliminary version of it was done in the lab, but it's still to be done to check whether this kind of theory, which allows you to predict the input-output relation for a sum of sine functions, actually leads to the correct results. Okay, so let me summarize what I've shown you here. First, that obviously, biophysical neuron models are complex and yet obviously very simplistic in a sense. And actually that within the framework we've discussed here, and stochasticity is essential for that framework, that additional complexity simplifies analysis. We generated excitability map for sparse stimulation, and through some assumption of linearization, we were able to obtain many quantities related to the statistics of the input-output map. One issue is to note the large parameter degeneracy. There are many neuronal models which will lead to the same kind of input-output of the input-output statistic relationship. And essentially, from our perspective, the analytic results we had here are essential to tuning the model, because there is no way you can ever tune a model with 50 parameters. You can always overfit, and this model has a far reduced number of parameters, and it's much easier to tune, and that requires these analytic results. Surprisingly, the approach works beyond the assumptions as I've shown you. We've provided at least one biophysical interpretation for this 1 over f behavior, and we're able to separate the contributions of the noise, the internal noise from the input, and demonstrated minute-long memory of single neurons. and the comparison with the experimental results, at least within the context of our model, our argument would be that many slow processes exist and that slow processes have to be noisier in a sense, that is, fewer ion channels. And the very, very slow processes affect the intrinsic noise, but less so the input-output relation. So just to conclude with a few open issues, as people very well know, there have been many models suggested for 1 over S, some stochastic, some deterministic. Not all these models are biophysically oriented, and essentially a deterministic model, for instance, or a statistical mechanics-oriented model has recently been suggested by Asaf Gal and Shimon Oron, which is based on the excitability fluctuations as reflecting critical phenomena and self-organized criticality phenomenon, and I think Shimon discussed that here. This model requires channel cooperativity. So I know this is a contentious issue, this issue of channel cooperativity, but if you allow for cooperation between channels, then there can be some kind of emergent effects of 1 over F within that framework, a deterministic framework. Now, moreover, we've made these linearization assumptions, and we know that often actually the actual chemical kinetics themselves are best described by nonlinear equations. So obviously, there are many issues to study, and I think from my perspective, the challenge would be to develop some kind of generalized framework which can combine stochasticity, cooperativity, and nonlinearity. The downside, of course, is this is a very difficult problem, but I think that at least we've shown that you can at least make one little step in that direction, and the interaction between all these processes is fundamental to understanding the behavior of even a single point neuron at extended time scales. Okay, so thank you.
Part of what Shimon talked about was a kind of entrainment factor, repeating the same stimulus and finding a reproducible outcome along.
Right. So that was, I mean, what he showed was that if you, depending on the statistics of the input, you can generate different types of correlations of the output with the input. And what they showed is that when the input statistics had this kind of 1 over F behavior, the correlation with the output was maximized. Now, we haven't looked at that in great detail, but it does seem to hold in our model as well. But it's something we haven't really tested very extensively. Yeah.
Your input was the sum of delta functions.
The input, yeah, essentially
What
fine.
happens if you start saying with the amplitude is generalized?
Oh, so what for? Each input has a different amplitude, or we haven't looked at that. I mean, the only thing we've done, which is kind of close to that, is that we let the inputs affect the neuron through synapses which undergo kind of depression process. So it essentially modifies their amplitude. Now, at least as far as the spectrum goes, we saw that it behaves pretty much the same. It doesn't have a dramatic effect, but this is something which we haven't really looked at in detail.
So the space of inputs that you are considering in a way, the dimension of the space of input is much lower than.
Well, it's just the variable that is just the intervals between So the intervals between the stimuli is the only dynamic variable we considered in terms of. We have looked at the effect of globally changing but not actually stochastically changing it.
Again, the 1 over S is a property of the input, not of the system.
Sorry?
Unless you have a strong nonlinearity in the system or very high dimension, I mean, you'll never get 1 over S unless the input itself has very high dimension.
Yeah, but you can get,
You have finite number of poles in your process.
Yeah, so if obviously you have a finite number, but you can actually get, if this finite number is sufficiently large, you can get a very, very good approximation to 1 over f behavior. Yeah, it's always because there is always a cutoff there, but you can have a relatively, I mean, I can show you a figure, but you can have a relatively small number of poles and generate something which looks very much like a 1 over f behavior, up to very reasonable time scales. Yeah.
You know how poles behave.
Well, actually, we have a figure of that in the paper, so I can show you. Yeah.
Any more questions? Okay, thank you very much.